Good morning, students. Today we will have to discuss about the post-fertilization changes in the plant. In fertilization, in previous classes we have discussed the fertilization. What happens during fertilization? After pollination, there is a pollen-pistil interaction, the formation of pollen tube, the movement of the both nuclei inside pollen tube, fertilization or fusion of the male gamete with the egg cell and secondary nucleus, and the formation of the pre-endospermic cell and pre-embryonic cell. After this process, what happens? The respiration will be increased and the ethylene formation will be also increased. That is, improve the respiration. Respiration rate will be increased and ethylene formation is also being increased. Both will be increased. Due to that, what happens? The calyx in most of the cases, corolla in all cases, anther and filament in all the cases, and stigma and style, these are disappeared. Only ovary become there and ovule is present inside ovary. Now we will have to discuss what happens after fertilization. You will see that this is the stigma, this one is the style, and this is the ovary. This one is the ovary. Inside ovary, there is ovule. There is ovule like that. This ovule have nucleus, have integument, and inside this nucleus there is a two-man conditioned pre-endospermic cell and a three-man conditioned pre-endospermic cell. It is called pre-embryonic cell. Three antipodal cells are remain and one synergid will be there. Now we will have to discuss that what are the fates of the different remaining parts, because during post-embryonic, post-fertilization changes, what happens? It becomes fall down, it becomes disappeared. First of all, what happens? These three cells become absorbed by the growing part of the nucleus. This also being absorbed is disappear. Only 1, 2 and remaining parts are present here. Ovary form fruit. Ovarian wall form pericarp. Ovule form seed. Integument form seed coat. Two-man cell, it is called pre embryonic cell and responsible to form embryo. And this 3N cell, it is called pre-endospermic cell. The fate of this particular 3N is to form endosperm. The fate of this particular 2N is to form the embryo. This integument responsible to form seed coat. The whole of this nucellus and this ovule is responsible to form seed, and whole of the ovary is responsible to form fruit. Now we'll have to discuss that how this endosperm is formed and how the embryo is being formed in the dicot and monocot plant. Endosperm. It is food-laden tissue in the gymnosperm. It is not 3N but N condition, but in the angiosperm it is 3N condition, and this endosperm is a food-laden tissue used in embryonic development. Used in embryonic development. Now, on the basis of the division of the pre-endospermic cell, it is of endosperm is of 3 types. Number 1, that is pre-nuclear endosperm. It has been observed that 3N conditioned cell, and this 3N is the nuclear part. It is mitotic division, and during this mitotic division, what happens? Each and every karyokinesis is followed by cytokinesis, and the cell wall is being formed. But in this condition, in pre-nuclear endosperm, what happens? The only nucleus are divided. Only nuclear division takes place, and this nuclei will be there. This is in the case of coconut milk. Coconut milk. Second type of division, second type of endosperm is the cellular endosperm. In this type of endosperm, each and every cell is completely divided into 2 cells, then to the 4 cells, then to the 8 cells, so on, and it will form a complete endosperm. Okay. Now, third one is the halobial type. Here it has been observed that firstly nucleuses are divided and they form many more nuclei. After that, it has been observed that it is surrounded by the cells having cytoplasm and cell wall well developed cell wall. So this part is called pre-nuclear endosperm and this part is called cellular endosperm, and this type of endosperm is called halovial endosperm, and it has been observed in case of it is coconut milk, and it is the edible white part of the coconut. So in coconut both type of the endosperm, that is called prenuclear and cellular are present, but this type of it is present in the, it is called halovial endosperm. The cellular endosperm is mainly present in the monocot plants. Now it has been observed that during seed formation, all the endosperms are exhausted. During embryonic development, it has been observed that the endosperm are not remains inside it. So it is called non-endospermic seed. Seed which is formed by this, that is called non-endospermic seed. But if the seed will be, the endosperm will be there after fully development of the endosperm embryo, then it is called peri-endospermic, or also called perispermic seed. This is the fate of that particular 3N condition, sir.